Good morning and good afternoon, everybody. I would like to welcome you to our webinar on sensory evaluation of foods, basic techniques. I am here together with my colleague Peter de Kock. My name is Kerstin Bursek. I'm a flavor scientist at NIZO Food Research. And my colleague Peter de Kock is principal scientist flavor. We are working for more than 7 years together in the flavor department of NIZO, and we are very delighted to have a nice discussion with you throughout the presentation. If you have any questions, please do not hesitate to send us your question. You can also send your question in Japanese. The questions in Japanese will be answered after the presentation, and normal questions that I could answer or Peter could answer in English, we will try to answer throughout the webinar. So please feel free to contact us with any questions related to the topics whenever you want. I will now hand over to my colleague Peter de Kock to give you an introduction to NIZO and also to the outlay of our presentation this morning.
Thank you very much, Kerstin. Thank you very much. First, I have some slides on NIZO Food Research. NIZO Food Research is an independent private contract research company, which means that effectively we do contract research for companies, most likely those subjects that they cannot directly solve themselves, and we help them innovate in the marketplace. It's a contract research institute that you actually see on the picture. This is our research facilities, and you can also see that we have a fairly large pilot plant. In fact, our pilot plant facilities are the largest free pilot plant facilities in Europe. And with free, we mean that anybody, any company actually can have access to this if they actually run a project with us. The whole institute is about 180 professionals, and one thing that actually makes it a bit unique is that everything we generate in terms of a project, it's IP, and the IP is immediately transferred to the paying customer, which means you. We don't have an obligation for publishing, so we can do actually research programs that are exclusive to you. In the next slides, you see what kind of services and deliverables we actually bring to the marketplace. One of our slogans, and you're going to see it in the remainder of the presentation, is actually together to the next level, and this is pretty core to our business. We are not doing this in isolations. We need you as a customer to work closely together in actually delivering the deliverables that we have been setting in the project. So what we typically do is we work together to deliver new processes, new formulations and recipes and product formulations, and we actually test these into production. Hence, we have the large pilot plant that you see on the right-hand side of this slide. Typically, we serve as an open innovation network for research, but we do quite a bit of consultancy in actually helping you setting a problem or defining a problem, and we do quite a bit of product and process development in making test products or prototype products, but also in generating in a kind of tolling operation setup the first products that are being launched into the market. In fact, also for clinical tests and intervention trials. Our expertise surrounds the core expertises which we located in the middle, in the gray area in the middle, which is typically application and production. And everything, I would say, is science-based. We come from a science understanding. We link into the information available from universities and other research institutes, and we bring this to application. And that indeed shows us that we're not only working together with our clients, but also with lots of academia. Now, this course is about the sensory evaluation of foods. This is the basic version. In a while, we're going to have an advanced webinar as well. But in this particular one, we're going to look with you to what actually sensory evaluation means for foods. So first we're going to start with an introduction to sensory science. So what is actually the role of sensory evaluation and why is it so important in food research? or setting actually your food, defining your foods and the quality they need to have. Quite importantly is of course the sensory techniques that are available. Why do they differ? Why are there different sensory tools available? And under which conditions do you have to apply these? And if you do sensory evaluation, you typically generate quite some data, and statistical methods for actually interpreting the data is quite crucial. Now, the human subjects in any kind of panel, they are not instruments. So you can have a readout in terms of the sensory evaluation that people have, but please realize that these are still human beings, and they respond not only, I would say, in analytical terms, but they always have a kind of emotion that is attached to any kind of food. We've been trying to visualize this in this plot, and you can see that although analytically the orange dots are of equivalent size in both graphs, if you measure them, the perception is quite different. Most of the people actually indicate that the right one seems to be bigger than the left one. And this means that any kind of sensory attribute is not being, have to taken into isolation, but it's an effect of all kinds of sensory attributes that surround the properties that you are studying. This means that perception is key to, I would say, interpreting the data that we have. Now, what you have to realize as well is that there's not only one sensory properties. If you assess a foodstuff, there's lots of different sensory properties actually kicking into place. So first of all, you have vision. There's a very interesting article from people from Nestlé that actually shows that within 50 milliseconds people decide whether a product is low or high fat. And they already doing this by just looking at it. So no tasting at all, no information other than just looking at the product. People already decide whether something is to be regarded high fat or low fat. Now sound is quite important. If you open a crispy pack, most of the people actually perceive this as a fresher product than if you have a dull packaging material. Next to this, there's of course mouthfeel. You can regard it as touch and sound in your oral cavity, and that's quite an important parameter which actually is being reflected in how fresh products are or how creamy products are, and that is actually affected by taste and smell as well. Typically taste and smell are perceptions that actually drive consumer preferences as well. So these sensory perceptions, they are being merged into your brain to some kind of hedonic response. Either you like it or you dislike it. Now there's a bit of difference between these sets of sensory senses. That means that texture typically with vision, touch and mouthfeel is actually being processed in your cortex, which is your conscious process. You can talk about this. Most of these things like the cortex, your visual cortex, is very close to your audio center, to your linguistic center, and you can easily talk about colors. For instance, we can easily discuss with people the color shadings in this bar, and people always agree that this is lighter blue, getting darker, and goes toward green. That's easy to talk about. Taste and smell is slightly different because it actually feeds into your limbic system, which is your most primitive part of your brain, and that doesn't really account for being able to talk about this. It's much more forwarded into your emotional response, and that actually makes taste and smell much more difficult to analytically, I would say, measure using a panel. People really have to be trained to recognize emotional responses for this, for a product, and that also has an effect, or in fact is being affected by the products that you actually make. So you can imagine that the products you actually make and prepare, the process to which it's being submitted, that is a key parameter in any kind of sensory attributes, and that is in fact being fed towards liking or a disliking reaction of your consumers. So typically what you see, you have a product sensory perception kind of cascade of information, and that actually makes it quite important to use sensory science and sensory scoring as a kind of intermediate instrument between your product characteristics on the left hand side and the sensory perception of consumers on the right hand side. Now for the role of sensory science, I switch to my colleague Kirsten Beck again, and she will show you what kind of analytical tools we have, and she will introduce also the first one of it.
Hello, it's me again. What Peter already nicely summarized that in sensory science we're using human subjects as instruments, and human perception is also very complex and very difficult to analytically describe. It is therefore very important that we choose the right techniques, the right subjects for the right tests that we want to conduct, but also to conduct the right data analysis. And many companies nowadays understand the need for sensory analysis, and they understand it costs money and costs time to conduct sensory analysis. But they also understand that sensory science really delivers a lot of useful insights to help guide product development and commercial strategies. But for instance, a new product development, if you want to launch a new product on the market, you really would like to know, will my product be successful or not? So you conduct consumer sciences. You ask people on the street, do you like your product? Don't they like your product? You, for example, want to consider a cost reduction or reformulation of your product, and you want to know, does it change somehow how the product is being perceived? Does it alter the appearance? Does it alter the sweetness? Does it alter the aroma of the product? And imagine you want to launch your product and you see it is not as good as your competitor product. What are the important attributes that you need to take care of to drive the liking and acceptability of your product? Furthermore, sensory sciences will help you to identify target consumer segments. It will help you to also to understand what is the difference between your product and a competitor product, and it also helps you to evaluate new product concepts. And finally, sensory sciences is also a very essential tool in quality assurance if you want to monitor the development of the flavor or the taste qualities of your food product during shelf life. So before you conduct a sensory experiment, you really have to be specific in what do you want to measure, what do you want to know about your product. Do you want to know, for example, a difference in sweetness if I change from a caloric sweetener to a non-caloric sweetener? Do I want to know if people like my product or not? Do I want to know if there's a difference between my product and a competitor product, and what kind of difference is it and how large is the difference? So based on your initial question, you will have to select the right test that you will conduct, but you will also have to select the right human subjects, the right assessors that you want to use. For example, if you want to ask for a very analytical description of the flavor properties of your food product, you are not using consumer people, but you are using highly trained expert panelists who are familiar with the flavor language, who are familiar with the evaluation of different sensory attributes. On the other hand, if you're interested to understand if your product is preferred compared to another product, or how much subjects or people like your product, you're going to use consumers, and consumers have to be naive. They are not trained because you want to have their innate response to your product. And finally, you also need to think about how I'm going to interpret the data. Sensory data is not necessarily parametric, so there are different tests, different statistical analyses that you have to apply to make sense of your data. I already outlined that the choice of the assessors is very important, and you also have to decide, do you want to train my assessors or not? If you have a product and you want to have an analytical evaluation by your subjects, you need to give them time to get familiar with the product, to get familiar with the sensory attributes. Then you do an extensive training of your human subjects. If you want to work with consumers, you're not training your subjects because you want to have their subjective response to your product. You also need to be aware that you are having a test location that facilitates the sensory evaluation. So it needs to be a room that is free of any smell, free of any noise. It should not distract the human subjects, and they should feel comfortable. And of course, very important is your experimental design. Depending on the test that you do, how many samples are you going to analyze? Keep in mind that human beings can only analyze a certain amount of samples in a certain period of time because of sensory fatigue and adaptation. So think about how many samples in a session you want to evaluate. Do I want to have replicates? In which order do I want to run analyze the samples? Usually we use fully randomized designs to avoid that any order effects or human subject influences will have a huge influence on the data outcome. And finally, you have to consider ethical points. For example, it is common that you have certain quality practices in your company, and keep in mind that when you're working with human beings, you have to guarantee that the samples are from a toxicological and microbiological point safe. Yeah, you don't want to poison your subjects, which is considered as unethical, obviously. You also have to consider that you need to protect the personal information that you obtain from your subjects. For some sensory studies, you look for certain consumer segments, so you get to know the age of the subjects, you get to know their habits and consumption, etc. All this data must never be available to a third person unless the identity of the human, of the subject is being hidden. Furthermore, you have to inform your subjects about the test to some degree, because you need to let them know what they have to expect, what they have to undergo through the test. You have to inform them about certain ingredients that might cause allergies, for example. If people are lactose intolerant, you should not give them milk-based products, and so on. And finally, you have to ask your subjects to sign a written consent that they understand what they are doing throughout the test and what potential risks are when they are conducting the sensory study with you. And finally, you need an assurance that covers any incidents that might happen where the subjects are being involved. It could be the travel to your place where you conduct the test. It can also be that during testing they slip and fall, so be aware that you have also a good insurance for your sensory people. So now I'm giving you a short overview about the different types of sensory testing that exist. So the most easy test is difference testing, and difference testing involves that you ask the subject, is there a sensory difference between product A and product B? For example, which of the two products is sweeter? Which of the two products is more moist? Which of the two products smells better or smells more like vanilla? You can also ask them how much the attribute differs between the two samples. So what is the difference in sweetness? What is exactly the size of the difference in vanilla flavor between the samples? And these tests are very straightforward. You usually only ask about one attribute, and here there's not necessarily a need to use your trained subjects, but you can also use untrained subjects. The second type of test is much more sophisticated. It's called Quantitative Descriptive Analyses, and here you evaluate all the sensory attributes that are important to characterize your food products. So you first do a qualitative analysis of the products, you make a list of all the attributes that are relevant, and then you give them scores. You ask the subjects to rate the intensity of these scores. Obviously, you will need a highly trained panel who understand what are different sensory attributes are that are known, that are very accurate in the way they are scoring the product. They're very reproducible in the way they are scoring, and they're obviously also sensitive and able to discriminate between odor and taste and texture properties. The final type of test is consumer testing, asking for consumer liking. Here obviously you're working with consumers, people from the street that are not trained, and you ask them to assess or to talk about the personal preference or the acceptance towards a certain product. I will now show you an example of difference testing. So I already outlined in difference testing, you want to know, is there a difference between product A and B? And usually it's recommended that you tell them, is there a difference? And they have to answer with yes and no. And you do not give them the choice to say, I don't know. Reason is that you need an answer. Is there a difference? Is there no difference? And if you ask for the maybe question, you might get confusing answers by your subjects because they are unsure how to score. They want to please you. They might not want to make a decision because they're afraid of making a mistake. So this is why in difference testing it is recommended to have a fourth choice, yes or no. In difference testing, you get a statistical significance whether there's a difference or not. There are certain statistical tools that you can use and tables where you can read if 10 out of 12 subjects say there's a difference, then I know it's statistically significant that actually my samples are different from each other. Difference testing is overall very rapid and easy to apply, and also very nice screening tools in quality development. It is recommended to have at least 24 human subjects if they are untrained for a difference testing. This is also to guarantee that you know that your difference is statistically significant or not. There's a question, and I will hand over to my colleague Peter de Kock.
Thank you, Kirsten. There's indeed a question from the audience, and the question is, if you prove there is no statistically significant difference, does that actually mean that the products are the same?
Thank you very much, Peter. Yes, indeed, it's a very good question. Sometimes you don't want to see a difference. Sometimes you want to have the answer that there's no difference. Imagine you want to reduce the sugar level in your food product for health reasons, and actually you don't want the subjects to perceive that there's a difference in sweetness. In this case, getting the answer there's no difference is the answer that you're looking for. Sometimes you're also happy if you observe a trend in your data. It does not need to be statistically significant, but for your own interest, you see there's a trend that there might be a difference, and that might encourage you to go further into detail and have a better look into your samples. So I hope I have answered the question, and if there's more information that you would like to know, then please contact me at the end of the webinar. Here's an answer. Here's an example of how you would conduct a difference test, and this type of difference test. There are different types of different tests, but this is the most common one. It's a true alternative fourth choice test or paired comparison test. And as I said, you can use trained panelists, but you can also untrained consumers. How do you conduct the test? You have the two samples that you want to evaluate. You have to present them in a blind manner. That means the subjects will not be able to differentiate the samples just by looking at them. That means you have them all in the same plastic cups. You have them all labeled with the 3-digit codes so that they get any distracting information about the sample. You asked the panelists only one question: which sample is, for example, more creamy, or which sample is more sweet? And then they have to indicate that it's either sample A or sample B on the screen. And then the panel leader, he's performing a statistical analysis to see whether the samples are significantly different or not different. In some cases, I already outlined you don't want to see it different. And then you get your probability value, how likely it is that there's a difference. For example, a probability value of 0.05 means that the chance that there's a difference, although there is no difference, is only 5%. And for example, if you have a total of 12 subjects that are conducting your test and 10 of them perceive there's a difference, then you can look it up on tables and telling you, yes, there's a significantly difference between my samples or not, if at least 10 of the test subjects say that there's a difference. And the result you are reporting in this way, you say the number of panelists who found sample X is more sweet, is this and this compared to sample Y, and then you state the significant level of significance that you found in your test. Be aware that if you conduct a difference test or two alternative choice tests, is that you only focus on one sensory attribute, not too many attributes at a time, because it's distracting and the data is suffering from quality when you ask too many attributes. Also be aware that you can only test one sample at a time against the reference. It's a paired comparison test. You have only two samples and you get the answer, there's a difference or there's no difference. If you want to compare more than one sample with your reference, you always have to give them as a new pair against your reference. And here is some data that we obtained at NIZO on the evaluation of the sweetness of a set of gels, and you can see that these gels all have the same level of sweetness. They have all about 10% sucrose, but we layered the gels, so we have layers where the sugar is homogeneously distributed, but you can also have layers with high and low levels of sucrose, and you can also differentiate how big is the difference between high and low. For example, you can have 15.5 or 20.0, 40 at the bottom, no sucrose here. You can have that there's a high sweetness reference, and then you have also a high sweetness reference where you layer 2 times 40% sucrose. And in this test we only asked the subjects, they compared the sample, for example this sample with this sample, and then they had to indicate which of the two do you perceive sweeter. Then as a next sample pair they compared this sample versus the reference, this sample versus a reference, this sample versus a reference, and this sample versus a reference. And here you can see the outcome of the paired comparison test. And in this, for the first sample you see that about the same amount of people say that sample A is sweeter than the reference, and there was no statistical significance, so they were basically perceived as equi-sweet. And if we are now looking at this sample, more people perceive this sample sweeter than the reference, and there you can see there's a statistical significance, that the statistics say that the difference between this sample and the reference in terms of sweetness is significant. It's also the same obviously for the sample with a higher sucrose level, and this sample here was perceived as the sweetest. So you can see it's a very straightforward test. It gives you very fast a straightforward answer if there a difference is or not, and also how reliable this difference is, and this is expressed by the probability value. Okay, I hear there is another question. Hang on, I will hand over to my colleague Peter again.
Thank you very much, Kerstin. Actually, there are a few questions. There are two questions which I will try to capture in one question. So you might not recognize your question, but it has to do with the difference between products and how many attributes people can distinguish between different attributes. So the question is largely, what is the number of attributes you can generate in setting difference or actually identifying difference between products? And is there is there something like a number of attributes that you use for this, or is it just products being different?
If I understand the answer correctly, in a difference test you can only ask one attribute, or you can ask more than one attribute in a difference test. It is recommended per session only ask for one attribute, and then conduct another session for another attribute. So obviously foods are very complex. You have a list of different texture, taste, and aroma attributes. But if you, for example, consider sweetness is the attribute that I want to focus on, you will only ask if there is a difference in sweetness. A difference in sweetness can also influence aroma, it can also influence liking, it can also influence the texture. This question should then be addressed in another test where you ask them, first, please tell me, is there a difference in sweetness? When you finish this question, go over to the next question, is there a difference in flavor, for example? So yes, you can rate more than one sensory attribute in a difference test, but they all be one after the other and not at the same time to not confuse the human subject. And now I'm going to hand over to Peter de Kock again. He will talk about quantitative descriptive analysis.
Thank you very much, Kirsten. I'm going to explain you what quantitative descriptive analysis is. It's a very powerful sensory technique, and this technique actually allows for different attributes. In most particular cases, when you have a difference test, you ask people to define a difference. You ask people to see whether two products are different in any kind of sensory attributes. They can focus, of course, on a certain attribute, but largely you see that when products are different, they can easily identify the difference, and they will always show a statistically the difference. If you want to know what kind of attribute is actually generating a difference, then quantitative descriptive analysis is a very powerful tool. In that, you allow actually a panel to describe or to rank or to rate a set of attributes which are all relevant for a certain product. So the sensory tool for using this uses sensory attributes, and of course these sensory attributes are product specific. Every product has a certain set of attributes that describes this whole environmental space which a product actually, or a set of products, occupies in, I would say, the whole sensory range of products. So any kind of product will need a set of attributes that is required to describe the sensory properties of that food. You really have to train panel on assessing a large number of these sensory attributes. They need to be trained not only in identification of this particular attribute, but also in its magnitude. So this is really a case where panelists allocate a value to a certain sensory attribute. So they come back with a score. If you have such a panel, and you can use them for many products, in fact. Again, every product will have its own set of attributes, but combined, this panel can actually cope with a large set of attributes with which they are familiar. And that means that in any kind of product, they will identify a set of attributes. They will need to specify the sensory properties of that product, but in fact they can have access to much more of these. They've been learning quite much more of these attributes which they can use for different products. So typically you can use a QDA panel for many different food applications. They're going to generate a scale. A scale is of course quite important in terms of its statistical interpretation. What you typically ask on a sensory attribute is to quantify it, most particularly in this example, as a product being not sweet at all to very sweet or extremely sweet. It's very important to give them a kind of reference sample as well. Sometimes we choose for two references, one at a low scale and one at a high scale. Sometimes we choose one in the middle. Very importantly is that, of course, you generate a score from a panel which is highly trained from a number of people. And it is quite important that these people are being trained not only on the type of attributes, but also indeed on the, I would say, the ranking or the number they actually generate. If you do not train these panels very well to identify a score to their references, you get extremely large amounts of noise in your sensory results. And noise actually translates into a low statistical significance. So then again, if you start setting up such a panel, please take care of excellent training, I would say, both instead of the types of attributes that the people actually agree on the identity or the identity or the quality of the attribute, as well as using the scale. Now let me give a bit more details to how this actually works. So typically you use a panel, a professional trained panel leader. There's somebody who is familiar with all the procedures that have to be set with using such a panel, how to work with people, how to get the best scores, how to motivate people. Motivation is extremely important for such a panel. And you actually need to generate a set of panelists of about 10 to 12. And in fact, there's a procedure for doing so. These are typically the top 10% of people, best tasters, I would say, in a certain region. And you actually select these from, I would say, the general population or from your company by a strict protocol that starts with identification of basic tastes, but it also has a particular element in, I would say, the creativity that people have, because this panel has to identify attributes themselves whenever they fall short. And that needs some kind of creativity for people to be able to identify new attributes which the panel has to be mastering in order to evaluate all samples. So the typical question is also, what will they test and how will they test it? As I mentioned, they will make a full list of attributes and they need to define consensus. Typically in these round table discussions, the whole panel gathers and they are actually tasting the products, sharing amongst themselves the attributes which they find relevant for this product, and they need to agree, all of them, they need to agree on what this sensory attribute actually means. And only then, after the whole panel agrees on this, I would say, attribute, then it's important to get some samples in making in giving it the correct ranking, as well as, I would say, the scaling of it. So you need to have a fixed baseline for scaling it against a reference or some reference samples. And in another session, the panelists will evaluate them individually and score them blinded. So once you have been set with a panel which knows the attributes, knows them how all the individual attributes can be ranked or scored against, only then you can offer them samples to be tested. And this is typically an environment that you see here where you do this kind of testing. So they're sitting in front of a PC screen where they can input their data, but as you see, all the booths are independently from each other, so that there is no, I would say, information exchange. And of course, next to that, you randomize all samples. So in the case that people actually make some kind of noise, even then it doesn't affect any kind of scoring. So the panel leader performs the statistical analysis. All these people actually generate scores for all the products, so it's a significant amount of data that comes out of it. And the panel leader actually does the statistical analysis and looks at the results in terms of the statistical significance of that. As you gather quite some data, you can statistically process them. So the results that you get is an attribute list, a score of all the samples on all attributes, and indeed also in a typical statistical analysis, which of the attributes vary significantly. And that's quite important as well, of course. ANOVA is an abbreviation which actually states analysis of variance, and that's actually the kind of trade-off between the noise in the panel and the differences in the scoring of each of the panel members for one sample, for instance, against what actually is the difference between all the samples. So it compares the noise in the panel to the variation it sees between all the products. Now, quite important is that the attribute list represents all attributes. Imagine that you submit a product to a set of people, but obviously not all the attributes they cover the whole sensory perception of a product. Then actually the panel gets confused because they taste something and they cannot allocate it to a score of a certain attribute. That needs to be avoided because actually two things can happen. One, and that's probably the best solution, is they will completely ignore this kind of sensory difference, so they will not score it. They will think, it's not in an attribute, so there's no way of me scoring it. The other thing that can happen is what we call halo dumping. That is that they notice some kind of difference or some kind of sensation. They cannot put it in a certain attribute, so they will actually merge it into different attributes or a single different attributes. And then actually you see that, I would say, the attribute list is being obscured by scores that actually do not belong to this specific attribute. You can avoid this a bit by also asking to make comments. So you can imagine if you give the panelists the opportunity of making comments after they scored all the attributes, then they at least know that they can actually put the information down on paper. So they are reassured that the information that they actually sensed is being processed in some kind of way. In the next slide, you see a typical attribute list. In this particular case, we prepared it for high protein sport bars. And in the sport bar, you can imagine that texture, for instance, is quite important. So there's a significant set of mouthfeel attributes, but that's of course not the only property that a sports bar actually has. It needs to have the right appearance, it needs to have the correct odor, but also handling is pretty important. People handle this particular product, and it needs to be firm, sticky, smooth, and for instance stable. Next to mouthfeel and afterfeel, this is the sensory element that kicks in after a while after mastication, and aftertaste as well. These are typical things that you can actually measure in this set of products. Again, this is an example only for protein sport bars, but you can imagine that many of the mouthfeel attributes, for instance things like creamy and spreadability, can also be used for many other products like mayonnaises and dressings and spreads. Now, using this set of attributes, running a QDA test, you get a set of scores, as I already mentioned. So on the next slide, you see such an example of these scores. You see a set of samples, arbitrarily labeled as sample 1 to sample 7. You see a set of attributes, and every one of them has a score. And these scores are in fact the averages of the panel members. So the first thing that you do is you take all the scores from all the attributes for a particular product, and you just make an average of all the scores. Now, that is not enough, of course, because you want to know whether a product on a certain attribute is statistically significant. And for that, as I explained already, we use an ANOVA analysis, which is a statistical procedure which is able to discriminate, or actually to indicate the relevance, the statistical relevance of these two products. Let me give an example on this slide. This ANOVA is being expressed in letters which is below any kind of combinations of a sample and a sensory attribute. For instance, the A in this particular example. This A means that this product is significantly different than a product labeled E. So any product with a different letter, it means that it is statistically different. When products have a letter in common, for instance A and ABC, then it means that this particular product is not statistically significant from this product, nor from anything that is labeled B or that is labeled C. It is statistically significant difference from E and also from C, D, and E. And so this is a very easy indication of showing the statistically significant difference between those products. Now you know that sample 1 on this attribute is significantly different from, for instance, sample 4. But the test was not so robust to actually discriminate on sample 2. It is also different on sample 5 because it doesn't have an A in common. So any product which has a letter in common is not statistically significant in its difference. Anything with a different letters actually is. So this is, for instance, on the score on the sensory attribute firmness. Now let's look at the second attribute, which is cleaning time. And you can imagine that cleaning time in a sports bar is, for instance, very important. How long it does take for a product to be able to be removed from your oral cavity. And in this particular example, you see that only sample 1 is significantly different from all other parts. So this clearly shows that product 1 actually stands out all the other products in this product set. And a third example, here you see stickiness, again quite an important attribute for a sports bar, for any kind of bar. You see that there is a set of letters indicating that this A, product 1, is significantly different from, for instance, this one and that one, but it's not significantly different. Also, for instance, product 2 is not significantly different from product 3 and product 4. And this means that you can also make a kind of ranking. On this, typically the sticky attribute, you can see that the most sticky one is product 1 and product 5. Product 1 and product 5, they both score an A. Then there's something intermediate, I would say, which are product 2, 3, 4, and 6. And the least sticky are products 3, 4, and 7. So product 3, 4, and 7. But you do see that they have overlap. Products 3 and 4, they belong to the category of medium as well as least stickiness. You can imagine that this is a continuous kind of scale that the panel uses to identify the stickiness of this particular product. So you see that you get quite some information from this, not only in terms of the scoring, but also in terms of the statistical variation within this data set. And it's not always easy to make a kind of overview of all the sensory testing that you've been doing. One very popular way of graphically representing all the data in such a test is with a spider plot. Here you see typically a spider plot of a product, of two products, a normal cheese and a cheese which has been specifically developed for a certain purpose. And in these radar plots, you see all the different attributes actually from the origin going to a higher score at the edge of the circles. So for instance, saltiness, it has a low score in the center and it goes up to a higher score at the very end of the scale. And that happens for all of the attributes. So this provides you a very easy, I would say, way of looking at your sensory data. It doesn't show anything on the statistical significance, but at least it gives you a very good overview of the scores. And you can very easily see that these two products are significantly different. For instance, this product is slightly more salt, whereas these products are more sweet and very much more brothy and also much more thermophilic. Now thermophilic will probably say nothing to you, but in cheeses it's a very important property. And the panel has been selected, or in fact has been trained to recognize this sensory attribute. And you can see that for characterizing cheeses, people have to learn and identify sensory attributes which are not common language, I would say, but this element is really needed to describe the sensory properties of all these products. And so you see in one, I would say, in a glance of an eye, you see immediately the difference between those products. But it gives only, I would say, little information. And if you have to compare dozens of products, for instance, you would have to prepare lots of these radar plots. And then again, this whole system actually falls short. There's another way of looking at this data, and that's typically by using what is called a PCA plot. Now, a PCA plot is in fact a statistical procedure which is a bit difficult to explain, and it's not the purpose of this webinar to go in depth in the statistical tools. But you can imagine that if you take a product, you rank it on a whole set of attributes, it's a kind of vector. Because you get a whole set of data on one product. And that's actually what's being shown here. Any product which is in blue, which is scattered all over the place, you can see there's many more products than the two that we had in the cheese example. It's actually an arrow going from the origin of this graph to some place in the XY plot. That also holds for the attributes. And so it's typically what you see here, you see a whole set of products with a whole set of sensory different attributes. And what this says, for instance, is that this product, which is product 3D, is very strong on spreadability. This product, for instance, which is 9E, is very strong on chewability. What you also see is that these errors are negatively correlated, because from the origin they go into completely opposite direction. And that means that on average within all these sets of products, things which are more spreadable are least chewable. And products which are very chewable seem to be very low in spreadability. So you see some kind of correlations between sensory attributes as well. Now you see also attributes that are almost perpendicular to this one. For instance, long and elastic. And that means, in fact, that there's no strict correlation between chewability and elasticness. And that, in fact, means that you can have a very elastic product being high in chewability, but also an elastic product which can be low on spreadability. So anything that is perpendicular, I would say, says that you have low correlations. And that not only shows, I would say, gives you a graphic representation of all your products, it also shows you what kind of how the sensory attributes correlate with amongst each other. Now what you see is a set of products which is typically a fudge kind of product, which is a kind of sweet, which is very long and elastic, and you can put it in your mouth and try to tear it apart, and products which are typically sweet like gummy bear, which are very chewable. And this comes out of a sensory plot of lots of combinations of all products which are typically in this kind of region of products. Now, what you can see is that these axes actually make a kind of sense, because this goes into elastic, long, this goes into firmness. So you can see what kind of attributes or what the principal axes in this PCA plots actually mean. This goes to grainy, from graininess to long, and this actually axis goes from sticky to firm. So it has some kind of sensory implications where the products actually lie, but now it's starting to make sense in terms of the physical properties of a product as well, because now you can understand how these axes come to existence and look into the products in how they actually how they are represented in this sensory plot. So for instance, what we've been doing, we've been grouping them accordingly, looking at this, how they have been prepared and what kind of thickening system they've been using. And then it started to make sense again, because this was typically a set of products which used a thickener which we arbitrarily called thickener A. This is a system which has a thickener system D in it. So it means that now they're starting to group apart depending on their ingredient composition. And that means that, for instance, if I'm using thickener A, it will make much longer and elastic structures than if I switch to a thickener D. And that, again, as I indicated already very early in the presentation, that is a crucial step of establishing what thickener systems actually do, composition of a product or the process of a product, actually what it does to the sensory properties of your product. So for the next slide, or for the next element of this sensory course, I will switch back to Kirsten, and she will introduce you to affective testing.
Hello, it's me again. We're coming to the last part of our webinar today, and I will talk about affective tests. And in affective tests, you ask for the preference or the liking of the product. So you're not asking for a specific sensory attribute, you're not asking for sweetness or creaminess, you just ask you like it or you don't like it, or you prefer my product over another product or not. So affective tests are usually used to assess the market potential of your product. Before you launch your new product, you want to know, will it be a success on the market or not, and you conduct a consumer study. And you can just ask people, do you like the product, yes or no? You can also compare your product versus another brand in the market, and then you see my product is as preferred or less preferred than the other product on the market. And if I say product, it can not only be food, it can also be a shampoo, it can be a toothpaste, a toothbrush, it can be any kind of product that is used by the consumer. And you also use affective tests and the final step in product development, for example, as a claim support. You develop a product which has 20% less fat compared to the regular product, but it's not perceivable by the consumer. So your claim would be it tastes as good as a high-fat version. And to be able to make this claim, you do consumer testing. You have them compare the two products, the high-fat and the low-fat product, and then tell me which one do you prefer. And if there's no actual preference between these two samples, so the same amount of people prefer the high-fat version and the same amount of people prefer the low-fat version, then, for example, you can have the claim tastes as good as the full-fat version. Affective tests are also very interesting to understand the preference of different consumer segments. So a consumer segment, for example, can be young people, old people, working people, unemployed people, people from a specific region, people who are vegetarians, people who don't eat eggs, etc. So it's also interesting to get to know the habits, the liking, and the preference of specific consumer segments. You can also conduct consumer testing to understand brand loyalties, for example. So how easy can you persuade a person to switch a brand, or how easy or how familiar are consumers with a product by comparing two different products that have no brand name, nothing on them. One is a standard version, one is a cheaper version. Do they actually perceive a difference between the two if there's no packaging around the two products? And you can also get an insight into what kind of products the consumers usually buy in the supermarket. For consumer testing, you should use not less than 100 subjects. Reason is that you are looking at people who are representative for the whole population. So you need a very huge number of subjects that are then representative for basically the rest of the consumer segment that you're looking for. And I also pointed out earlier, when you do consumer testing, the subjects are not trained. You really want to have their innate, their personal response, a subjective response towards the products. Where do you conduct consumer tests or affective tests? There are different possibilities. First of all, you can conduct the test in your laboratory. There you can really control the environment. You can control how the subjects, the consumers, consume the samples, how they conduct the test, etc. But on the other hand, you're taking them out of their normal environment. They might feel uneasy, uncomfortable, and it's maybe also in some cases not possible to do it in the lab. For example, if you want to test a shower gel, people have to do it at home in their own shower. You can also conduct central location tests. This is you going to a place where you know you will find meet the right consumers that you are looking for. For example, you're going to a certain supermarket and you give them free samples of your chocolate, and then you also ask them to tell them how much they like your chocolate or not. And finally, as I already pointed out, you can have home use tests. That is, you give the consumers instructions and the products to test them at home. For example, over 1 week they have to eat in the morning a certain yogurt, and then they have to tell you every day how much they like it, and you're interested to know if their liking increases over the time of consumption or not. Or if you, for example, look at cosmetic products, they are now asked to use your toothbrush for 1 month and then give you an indication, will they continue using this toothbrush, or maybe they don't like it at all. So these are tests that you usually do then at home, and the disadvantage is you cannot control if they actually do the test or just fill in any numbers on the scoring sheet. But on the other hand, you have the possibility to test your products under normal conditions. Affective tests can be divided into 2 different types of tests: preference tests and acceptance tests. And preference tests is basically you ask them to make a choice. You ask them which of the 2 samples do you prefer, or you can ask them which of the 2 samples do you like better. And an acceptance test, you also ask them to make a quantitative stating. So it's a rating. So how much do you like the product? On how acceptable is your product? For example, on a scale from 9 to 10, tell me how much you like the product. Is it an 8, then you're happy? Is it a 2? Obviously, you're not very happy. And here's an example for a standard test, for a paired preference test. where you ask for the choice between two products. And examples where you conduct such a preference test is you want to launch a new product and you want to know, is your product compared towards a competitor's product. Another example for a paired preference test is you want to reduce the costs of the product, the cost for the production of your product, and you want to compare the product with a new reduced formulation towards the common product, and then you can also ask, do you prefer one of the two, the new one or the old one, and ideally there will not be any preference for one of the two. You can also, as I already said, use consumer testing if you want to make a claim that your product is better than, for example, the market leader. You develop a new shampoo which is much better, it gives you much more volume for your hair compared to a competitor product, and you conduct preference testing, and 90% of the consumers you ask say that your product is much better than the product of the competitor. Then you can also use this result as a claim on your packaging. This is an example of how you can rate the acceptance of a product. As I said already earlier, you can also get an indication of the magnitude how much subjects like your product. So you can ask for preference and you get the answer yes or no, or you can ask for how much they like something, and then you can get kind of a quantitative indication. And this is a so-called 9-point hedonic scale. It goes from I like this product extremely to I dislike this product extremely, and then you ask them to test the product and also then rate them how much they like it on this scale. For example, in this case, somebody said, yeah, it's quite okay, I like it moderately. Yeah, it's not the best, but yeah, it's doing fine. How do you analyze the data? So you can convert these verbal indications into numbers and then calculate the average. For example, it usually scored very higher product, and your average would be like it very much, and that would be in numerical terms an 8. And then you can also conduct statistical analysis to see if it is significantly different towards a second product, for example, where you only get dislike very much. But you have to be very careful because these verbal indicators are not true numbers. This is not parametric data, and you don't know that the differences between the different indicators are not necessarily the same. For example, before people rate something as I like it extremely or I like it very much, there might be a huge perceptual difference between these two indicators, but the decision to make your cross between dislike slightly and dislike moderately, that distance might be much smaller. So it's not really that you can say there's an equal distance between the different verbal indicators, and therefore it's recommended to use non-parametric tests to analyze the data. You can also use the so-called just about right scales. It's a bipolar scale. It says too sweet or not sweet enough, or it can also be just about right, your product, and then again your consumers, they test the product and they have to give an indication what they think about the product. In this case, yeah, it's between too sweet and just about right, so yeah, it's doing okay. So the downside of the just about right scales is that subjects assess the intensity of the attribute, in this case sweetness, relative to a mental criterion. And we had this also in the discussions earlier throughout the webinar when I was answering questions. Yes, consumers vary a lot. Consumers have very different sensitivities, expectations, experiences, and all this information is influencing the data, the way they are scoring the samples. And the just about right scales, what is actually just about right? For example, I like things very sweet, so a very sweet sample for me would be just about right. For somebody, it would be too sweet. So again, be aware you're doing consumer analysis and you have untrained people and you have a subjective response towards whatever you're asking them. The benefit of consumer sciences is that you really get an indication of population, yeah? So, and you want to know, is my product liked or not? Test a lot of consumers, and then you can also see that you can split the number of subjects into different groups, and there are groups of people who like your product a lot, there are groups of people who do not like your product. And then you can make a choice and say, okay, I see that women age of 35, they like my product very much, but men at the age of 20, they don't like it a lot. So I decide my target group could be then women 35, and I produce my samples only for them. So I found my target group basically also by using the just about right scales. How do you analyze the data from the just about right scales? Well, you can analyze the percentages of how much, for example, too sweet was ticked, how much not sweet enough was ticked by the subjects, and then you can compare the data with a reference product. For example, here the product in green was much more preferred, or was rated as much more right compared to the sample in red. So this also gives you an indication of how well does my sample perform towards another sample. So I'm now coming to the end of our webinar, and I just give you a very short summary. If you want to do sensory analysis, be aware that you're choosing the right panelists, that you're using the right kind of sensory test, that you are aware of what you want to do. Do you want to just see, is there a difference between my samples? Do you really want to know what the differences are? You can also do more advanced techniques. For example, how does a certain attribute perform in time? This is time intensity or temporal dominance of sensations, and those topics will be addressed in our following webinar. And it's also very important that when you have your data collected, that you also use the right data analysis in the data interpretation. Okay, so the final slide. This webinar, we try to point out that sensory testing is really crucial for connecting technical product specifications to consumer liking. You have seen that there's a range of different sensory tools available, and every sensory tool has its own purpose. So make a good choice depending on what you want to know about your product. Choose the right test, the right tool. Also, every sensory test requires a dedicated panel. You have the highly expert panel who are highly trained and screened for sensitivity. If you want to have analytical data, for example, if you really want to know what are the key attributes of my product, next to only just I like it, but why, for example, if you want to understand why consumers like it, you can link your consumer data to quantitative data with the expert panel, who then make a nice overview of all the attributes that are relevant for the food product plus indication. And what Peter explained to you, it's a nice way of linking consumer data and the actual product qualities. And finally, be aware that whatever the training is you do with your panelists, they are still humans. Peter already mentioned at the beginning, there is multisensory integration. Even if you only ask for sweetness perception can be influenced by the smell, vice versa, the smell can be influenced by the taste. The way a product looks like can also influence your rating. For example, if you have white wine and you dye it red, people will name it and perceive it as a red wine, although except for the color, the taste and the flavor attributes are the ones of the white wine. So be aware subjects can easily be deceived, and be aware that you give them as little information about the product as possible to not bias them. For example, remove any packaging, label them with a 3-digit number. Don't call them sample 1, 2, or 3, because it already might indicate that sample 1 is better than sample 2, etc. So yeah, human beings are not instruments in the end. And yeah, I hope you enjoyed the webinar today and you learned a lot. I tried to answer your questions as much as possible while Peter was talking, but I'm very happy to answer any other kind of question that you still have, maybe some things that were not clear or you would like to have more detail about certain topics that we addressed in the webinar. Please do not hesitate to send us an email. We are very happy to answer these questions. As I already mentioned earlier, you can also send these answers in Japanese, like my colleague Liz. She is fluent in Japanese, so she will do her best to also address questions in Japanese. And then on the 18th of July, we have our next webinar, which is then covering advanced sensory techniques. And here we will also introduce techniques where we basically hook up human subjects to instruments. This is very important if you want to, in product development, if you want to have a very defined stimulus and you want to measure how tiny changes in the stimulus concentration affect the overall perception of a food product. But these are all things that you will learn in the next webinar. And there will also be more sophisticated sensory techniques, for example, time intensity measurements and temporal dominance of sensations, which is addressing how a sensory attribute is evaluated in time. We also have exciting techniques like PTRMS, where you actually measure the release of volatiles in the oral cavity that are transported to the nose while you're consuming a food product. And we also show you olfactometer and gustometer techniques that we use to, for example, measure cross-model integration. I thank you very much for your presence, and I hope you enjoyed it as much as we did, and we are looking forward to welcoming you to our upcoming webinar on the 18th of July. Bye-bye.